Hello and welcome to my YouTube channel. This is Dr. Bijoya Chatterjee. Today's topic is oncogenes, tumor suppressor genes, and cell cycle. This is in continuation with lecture series on cell cycle. Let's proceed. The specific learning objectives of today's lecture is to learn about proto-oncogenes and oncogenes, to learn about tumor suppressor genes, and to learn about the regulation of cell cycle by two tumor suppressor proteins, that is the retinoblastoma tumor suppressor protein and p53 tumor suppressor protein. So let's start the lecture with oncogenes and proto-oncogenes. Oncogenes are genes that are capable of causing cancer. The inactive form of oncogenes that are present in the normal cells are called proto-oncogenes. The proto-oncogenes code for proteins that stimulate cell division, cell differentiation, and regulation of cell death. So that is the difference between oncogenes and proto-oncogenes. Some cellular oncogenes are tabulated here. Let us take a look at those examples. Oncogene ABL is associated with human neoplasia chronic myelogenous leukemia. AB is associated with breast carcinoma. MYC is associated with small cell carcinoma of the lung and Burkitt's lymphoma. RAS gene is associated with squamous cell carcinoma, urinary bladder carcinoma, lung carcinoma. So these are some of the oncogenes, the names of which you are expected to remember. Oncogene activation. So let us see now how these oncogenes are activated. The change from a normal proto-oncogene to a cancer-causing oncogene is called oncogene activation. Oncogene activation is done by two ways. A. Activation of oncogene by viruses, and B. By mutation of normal cellular gene. So let us first take a look at the different oncogenic viruses. These are currently 7 viruses known, and these are known to cause cancer. Therefore, they are technically called as oncogenic viruses. The example of the viruses and the cancer that they cause are tabulated here. Let us see. The human papilloma virus causes cervical carcinoma. Epstein-Barr virus causes Hodgkin's lymphoma. Human T lymphotrophic virus causes adult T leukemia, T cell leukemia. Kaposi's sarcoma is associated with Kaposi's sarcoma. Merkel cell lymphoma virus is associated with Merkel cell carcinoma. Hepatitis B virus and hepatitis C virus both are associated with hepatocellular carcinoma. The reference for this table is given below. Now let us see how these oncogenic viruses act. The genome of viral DNA is very small. Now when a virus infects a host cell, its DNA goes and attaches to the host DNA, and it tries to incorporate itself into the host DNA. Once this has been achieved successfully, the DNA viral integration into the host genome has occurred, and the products of this recombinant DNA produces various viral proteins which cause cancer. Oncogenes and oncogens are different. Now these are two similar sounding words. Let us see what is the difference. Oncogenes are chemicals that produce cancer, whereas oncogene is a gene causing cancer. So oncogens are cancer causing chemicals and oncogene is the gene causing cancer. Oncosuppressor genes. So after knowing what are oncogenes, proto-oncogenes and oncogenic viruses, let us look at Suppressor genes. These are also known as tumor suppressor genes. That is the alternative name. The genes that code for negative regulator proteins that prevent the cell from uncontrolled cell division on activation are known as oncosuppressor genes. The function of the tumor suppressor gene protein is to put a roadblock to cell cycle progress until certain events are completed. So these proteins, these tumor suppressor gene proteins, do not allow the cell cycle to progress until certain criteria are met. A cell that carries mutated form of these tumor suppressor proteins or negative regulators, or if the gene of these proteins is deleted, it results into cancer. The important oncosuppressor genes include gene for protein p53, retinoblastoma, Wilms tumor, familial adenomatous polyposis deleted in colon cancer, and familial breast cancer. These are some very important examples of oncosuppressor genes, and you are expected to remember the names of these genes. In this lecture, we will be discussing in detail about the role of protein 53 and retinoblastoma in cell cycle. So we will see how these two proteins regulate the cell cycle. Now this is a normal cell, and when the cancer causing agents, whether it is biological in form of virus or it is any chemical or UV rays that affect the nucleus of the cell by activating the proto-oncogene. Once this proto-oncogene is activated, it is converted to activated oncogene, and the cell undergoes uncontrolled cell division and it becomes a cancer cell. Now we will see the cell cycle regulation at different checkpoints. So we know that there are two main checkpoints at which the cell cycle is regulated, that is the G1S checkpoint and the G2M checkpoint. So we will see the regulation at these two checkpoints, and the proteins that carry out this regulation, that is the retinoblastoma protein and p53 protein. We will take a look at that. Tumor suppressor genes or the retinoblastoma gene. It is the first oncogene to be identified. RB gene encodes for retinoblastoma protein. It is known as the guardian of G1 as it regulates the passage of cell from G1 to S phase. It suppresses cell proliferation, prevents activity of various oncogenes, and so it does not allow the cell to enter into the next phase from G1 to S, and therefore it is known as the guardian of G1 checkpoint. Now this is the long arm of chromosome 13, and you can see that this chromosome has the gene for retinoblastoma protein. These genes express themselves and the RB protein is formed. This is a normal retinal cell. When one of the chromosomes is affected, that is the retinoblastoma locus is deleted, still the RB protein is produced, but the retinal cell is at risk. Now, if the RB locus in both the chromosomes is deleted, it being a recessive trait, retinoblastoma is expressed in the retinal cell. So what happens is that a healthy eye um, is now converted to a retinoblastoma because a tumor is formed due to the missing retinoblastoma gene, and this tumor causes blindness, and this is how the RB gene was found. Now let us take a look at the regulation at G1S checkpoint that is carried out by the retinoblastoma protein. So let us see what exactly retinoblastoma protein does. In normal conditions, in a cell, the retinoblastoma protein binds to this transcription elongation factor E2F in the eukaryotic cell and inactivates it. So this is an inactivated condition. So what happens is that it blocks the transcription and prevents the cell from entering into the G1 phase. That is, the cell is held at the G1 checkpoint, so it cannot go into the next phase, and this is possible because the retinoblastoma is dephosphorylated, and in dephosphorylated condition RB protein is active, and there is G1 arrest, there is no transcription. This is what is happening in a normal cell. Once various factors like cyclin D, cyclin E, and different cyclin dependent kinases like CDK2, 4, 6, and various growth factors are produced or synthesized, the transcription can proceed. So let us see what happens then in controlled cell cycle, when the cell cycles or cell divisions are controlled. Once all the requirements are fulfilled, the G1 arrest is removed. So let us see how that is done. Now this is the retinoblastoma protein. In active stage, it is dephosphorylated and cell division stops. But when the cell is ready to enter the next phase, this retinoblastoma is phosphorylated. Once it is phosphorylated by the different cyclin CDK complexes. Look at the cyclin CDK complex here. It is cyclin D1, which is complexed with CDK4 or CDK6. This complex phosphorylates the retinoblastoma protein, and on phosphorylation, these two separate out and E2F is released. It is free to carry out transcription now. And once the transcription is initiated, the CDK6 leaves the nucleus, and this ensures that the the cell DNA synthesis does not occur at the same site. So initiation is at the nucleus remains controlled. And there is one more protein over here, that is the P16 protein. The role of this protein is to suppress the tumors and regulate cell cycle by inhibiting the CDKs. So whenever the CDKs are inhibited, then the cell cycle is arrested. Currently in this condition, when all conditions are fulfilled, G1S movement occurs and the transcription starts. Now we go to the dysregulation of G1S checkpoint. What we saw in the previous slide was what was happening in normal cells. Now what exactly happens in the cells when they have lost the G1S checkpoint control? So all cancer cells show dysregulation of G1S checkpoint. Mutations may be caused by one of the key regulators. It can be 16 CDK inhibitor loss function, cyclin D excessive production, CDK4 excessive function, retinoblastoma loss of function. So these can be one of the causes of dysregulation of G1S checkpoint. Normal cells in this case are transformed into cancerous cells. Now we will look at the role of retinoblastoma. So in a non-proliferating or non-dividing cell, what is happening? There are growth inhibitors TGF beta. It stimulates CDK inhibitor P16, which we saw. This P16 inactivates, and therefore cyclin D CDK4/6 or cyclin E CDK2 complex is inactivated. So retinoblastoma is unphosphorylated and it binds to this E2F factor, and therefore in this dephosphorylated condition the transcriptional block is there and the cell cycle arrest is existing at G1S checkpoint. So this is how the G1S checkpoint is regulated in the non-proliferating cells. Now what happens in proliferating cells? The retinoblastoma E2F factor are in complex form. In normal condition, there is release of growth factors EGF and PGDF, that is epidermal growth factor and platelet derived growth factor at the early G1 phase, and because of this the cyclin D CDK4 CDK6 complex is formed at the late G1 phase, and there is also formation of cyclin E and CDK2 complex. These two cyclins are able to phosphorylate retinoblastoma protein. Once it is phosphorylated, it separates from the E2F factor, that is the elongation factor, and now this elongation 2 factor 2F is free to carry out transcription. So it initiates transcription and there is activation. So this is what is happening. Tumor suppressor gene, the TP53, which is also known as the guardian of genome. Now we already know that the tumor suppressor gene is located on chromosome 17 and it is responsible for a tumor suppressor protein known as the P53 protein. Let us see what is its role in cell cycle regulation. So it is expressed in all the cells. It plays a vital role during cell cycle just before replication in S phase, and it regulates G1S phase of cell cycle progression and DNA damage monitoring. If the damage cannot be repaired, it causes apoptosis of the cell. Mutation of the P53 gene is almost common in all spontaneous cancer. The P53 gene is a regulator for both G1S checkpoint as well as G2M checkpoint, and the T53 gene provides instructions for making a protein known as the tumor protein P53. This protein acts as a tumor suppressor and it controls cell cycle arrest, DNA damage, senescence, apoptosis. So through these controls, it guards the cell cycle and keeps it in regulation. The P53 suppresses neoplastic transformations of cells by these mechanisms. Let us look into the details of this. There is one protein known as the MDM2 protein which regulates the P53 levels in normal cells. So now we know that the P53 regulates the cell cycle and the level of P53 is regulated by MDM2 protein. The G2M checkpoint regulation is carried out by cyclin B CDK1 complex when it is in phosphorylated condition. Because in this phosphorylated condition, this complex is inactive and the enzyme phosphatase then removes this phosphate group, and so this complex is now dephosphorylated and hence it is active. On becoming active, it phosphorylates several proteins which cause the spindle formation to occur properly, and this leads to mitosis and the cell divides in a proper regulated manner. When there is DNA damage, the protein kinase enzymes are activated due to this damage, and this will not allow the phosphatase enzyme to activate the cyclin B CDK complex. Because of this cyclin B CDK complex remains phosphorylated and so it is inactive. Because it is inactive, the phosphorylation of several proteins required for spindle formation are not carried out, and therefore this leads to cell cycle arrest at G2M. And this is how the G2M checkpoint regulation is broken due to DNA damage. In my previous slides, I have mentioned that P53 protein is known as the guardian of genome. So let's try to understand why it is known as the guardian. P53 gene protein is a 53 kilodalton protein which stabilizes the cell at G1S and G2M checkpoints. The increased P53 protein delays the progression of cell cycle through both these checkpoints. It delays the cell cycle progression, and this delay allows the time for DNA to repair itself if there is any damage. In severely damaged cells, P53 induces cell apoptosis so that the replication of damaged DNA is prevented. So, in all, it prevents the DNA damage from being transmitted into the next generation of cells. Hence, it is known as the guardian of genome. Let us see how the P53 protein acts as a guardian. Now, whenever there is a normal cell, the P53 feedback regulation for its activity exists. Increased P53 induces another gene which makes MDM2 protein. This MDM2 protein is also known as murine double minute 2 transcription protein. This protein is a ubiquitin ligase, so it induces P53 degradation by ubiquitination. Once this P53 undergoes ubiquitination, the proteosomes degrade the P53 protein. This occurs in normal cell. Whenever there is stress, the P53 protein undergoes phosphorylation, and on phosphorylation it becomes active. It goes and sits on the target gene and various proteins are produced which go and suppress the cell cycle, and this is how the cell cycle is put to a halt so that first the DNA can undergo repair. This is how the P53 acts. Thank you for watching. Like, comment and share and subscribe to my channel.